It's a pleasure to introduce Tom Newson-Davis, Rohit Lal, and James Spicer to discuss the latest developments in lung cancer.
So at this year's ASCO, there were a number of studies addressing some new drugs used in new settings in lung cancer, and I'll try and cover a few of those now. Perhaps the most high profile at this meeting has been this drug, Ganetespib, which is a new generation HSP90 inhibitor. So HSP90 inhibitors are nothing new, and galdanomycin and its related drugs, things like 17-AAG, have been around for a while but have not had a great impact from an efficacy point of view, and certainly are not terribly nice to use from a toxicity perspective. This drug perhaps seems a little bit different and is structurally quite distinct from those drugs, and as we'll hear, has a more acceptable toxicity profile. So this was the previously reported single-agent activity, a waterfall plot for this drug. So a downward deflection on the right here, it represents those patients responding to single-agent therapy. And really I put this in here just to highlight some of the molecular subsets that seemed perhaps to be important for this drug. In particular, you can see that those responding rather strikingly seem to correlate with patients harboring an ALK translocation. So actually when we come to discuss this randomized trial, you'll see that that was taken into account. So as is often the case in lung cancer, trying to put a new targeted therapy into the clinic, the first step is often combining with single-agent docetaxel, one of the very widely used therapies, and this is some preclinical data supporting the synergy for these two agents, which both between them seem to target the microtubule and G2M cell cycle progression. So this is a randomized phase 2 trial, relatively modest study size-wise, with about 125 patients in each arm. And the concept was to explore efficacy, in particular in some of those molecularly defined subgroups. In fact, the co-primary endpoint was PFS in KRAS mutated and elevated LDH patients. And patients were randomized either to docetaxel full dose or to Ganetespib in addition to docetaxel, so docetaxel full dose in both arms. So here's the toxicity, and I think the take-home message here, a lot of numbers, but considering that we're using full dose docetaxel in the experimental arm and adding in an extra drug, the toxicity was reasonably manageable. Certainly there was more diarrhea, certainly there was more fatigue and more febrile neutropenia in the experimental arm. Efficacy results. So PFS, there was a statistically significant improvement in the whole patient population. This is just adenocarcinoma patients. We might discuss why that was at the end of this, because at the beginning of the trial it was all non-small cell lung cancers that were being recruited. So statistically significant, but perhaps not very clinically significant benefit there, and overall survival, which was not significantly different. But then what was really focused on in the presentation here at ASCO was the progression-free survival, which was analysed prospectively in stratified populations, either progressing after or less than 6 months following diagnosis. So again, a slightly controversial aspect to this study, which generated a significant amount of discussion. But it's not wholly without precedent, this approach, and it was pre-planned. And the PFS benefit was 2 months, statistically very significant, and perhaps most importantly, an overall survival benefit of more than 4 months, statistically significant, was seen in this group. The mutation and other biomarker selected patient analysis was not presented at this meeting, as that data is not mature. So the key messages from this trial, this was a Ganetespib in combination with docetaxel versus docetaxel alone study, and it showed certainly in the subgroup who had progressed more than 6 months after diagnosis, both an improved overall survival and progression-free survival compared with chemotherapy alone. It's worth pointing out that the phase 3 trial is already ongoing and will be recruiting at a number of sites in the UK. It's an international study, and patients are being selected according to time since diagnosis, so they have to be progressing at more than 6 months since diagnosis. So moving on and changing tack, this is a monoclonal antibody approach targeting PD-1 and its interaction with PD-L1. Those attending this ASCO really couldn't fail to notice the impact of this class of drugs now. Many pharma companies have an antibody targeting either the receptor or its ligand, and after data presented and published last year suggesting that really quite striking efficacy in a number of tumor types with early hints about lung cancer, this was selected as an oral therapy, I think because this is quite a large group of patients now, albeit non-randomized, actually in a phase 1 trial, 52 patients with non-small cell lung cancer who had been treated as part of this study. So if you're interested in the phase 1 data, that was presented also at this meeting by Roy Herbst, but this is a separate presentation just of the lung cancer patients. Patients were included irrespective of their PD-L1 expression. So this is an antibody targeting the ligand, which is expressed potentially on many tumors. And of course, this was a phase 1 study, so the primary objectives were safety with a secondary efficacy endpoint, which was presented. So here is the safety data. Perhaps most strikingly compared with some of the other agents in this class which target PD-1 rather than the ligand, we did not see any grade 3 to 5 pneumonitis or diarrhea. So this is phase 1 data, and I think it's too early to claim that this approach is more or less safe than PD-1 targeting, but that's certainly an hypothesis. Dissecting out some of the subgroups, so in the top row of this column, overall population, that's referring to the fact that this was a phase 1 trial, albeit a very big one, 140 patients in many tumor types. But for the patients that were included in this efficacy analysis, there were 41 non-small cell lung cancer patients in that analysis, and the response rate was a very respectable single agent, 22% in pre-treated patients, and some nice maintained stable disease. Some suggestion perhaps that the squamous patients relatively benefit. And then at the bottom of this slide, an analysis of response according to expression of PD-L1. Now that begs the question, how do you actually measure expression of PD-L1? And this is a bit of a heart sink because it's yet another proprietary company-developed co-diagnostic immunohistochemistry test, but all the same, perhaps a bit of a cross to bear for us in the clinic for the future. But focusing then on the data on the last row of this slide, of the small group of patients who are PD-L1 positive, very high response rate, and for those who are ligand negative, a reasonable, not zero response rate. So one of the questions outstanding is exactly how do we use this diagnostic, and should we be excluding patients who don't express the ligand from treatment? Perhaps not, according to this data. So those were the newer drugs included in the main oral session at this ASCO for lung cancer. There were some other interesting developments presented in other parallel oral sessions, this one in a special targeted therapy symposium. So this is a BRAF targeting approach, dabrafenib, which is GSK's drug, very recently, only last month, approved for melanoma. And these are patients selected according to BRAF mutation, very small single-arm phase 2 trial from France. Interestingly, it was very specifically the V600E mutation which was required for participation in this trial, there are a number of other BRAF mutations which drive lung cancers, and in fact V600E represents only about half of all BRAF mutations, so we're talking about a subgroup of a subgroup here, and how we actually identify these patients is one of the main issues. So it was a two-stage phase 2 study, and efficacy in the first stage meant that they went on to recruit the full number of patients. So moving on to the efficacy for this for dabrafenib in these selected patients. These are the first 20 patients in the two-stage trial. You can see that there was a very healthy confirmed response rate of 40%. So the key message from this trial is, and I mean this is impactful because it's really the first strong signal of efficacy for a BRAF inhibition approach in non-small cell lung cancer, and certainly for dabrafenib. If you can find the patients, it seems like this may be a way to go. Certainly a reasonable safety signal, nothing new compared with what we know from melanoma. Now the way forward for this drug and this trial is to expand the sample size and extend the patient selection into first line. So that concludes our review of some of the highlights of newer drugs in the lung cancer track here at ASCO, and we'll have a discussion with some of my colleagues about that in a minute.
Thanks very much, James. That's a really good insight into compounds that are moving forward into mature clinical trials. What can you tell me about the way that we assess tumour with this new class of compounds? The anti-PD and the anti-PD-L1 group have an immunotherapeutic effect.
Yeah, so what you're referring, I mean, I think that's a good question, and because this was a phase 1 trial rather than one of the custom-designed later phase trials that we'll be increasingly hearing about with this class of drug, the actual RECIST that was used was conventional, but we should say a few words about the RECIST modification that's generally used in immunotherapy studies now. And the reason for that is that if we think about the way these drugs work, they're designed to switch on the immune system and bring in immune effector cells into the tumour. So what we certainly see with these sorts of drugs is in some patients in the early weeks of therapy, actually the tumour might be getting bigger, despite the fact that the patient then, if the investigators hold their nerve and keep the treatment on study, they then go on to benefit. So it does have some very real everyday concerns in treating patients in these sorts of trials, that we're identifying those flares appropriately and not discontinuing in patients who are going to benefit. But on the flip side, that we're not hoping in vain that somebody's progressive disease is just a flare. So that's something that as these drugs, as they inevitably will reach a standard of care in some diseases and quite possibly in lung cancer, given current evidence, we will, as a community, have to relearn how to use RECIST.
And in fact, some of the patients developed new lesions whilst they were on drug, but continued to gain clinical benefit.
Yeah, well, I mean, I think that shouldn't be a surprise to us in this era where everybody's talking about tumour heterogeneity, and that has to apply, I think, equally to the immune phenotype of a tumour, just as to the expression of TKI targetable driver mutations, for example.
Let's ask you about the BRAF study. The mutations we see in lung cancer, how are they similar and how are they different to the ones that we're seeing in melanoma?
Okay, so BRAF is a tricky customer because perhaps it's unlike KRAS, certainly unlike EGFR, there is really rather a smorgasbord of mutations that we see in this, in various tumours. In melanoma, it's relatively straightforward because there is less heterogeneity than we see in lung cancer. In lung cancer, as I mentioned in the presentation a minute ago, there is really a broad spread. So the V600E mutation, which was targeted very selectively in this French trial, really represents only about half of the spectrum of mutations that we see in lung cancer. So that's going to be an issue. it's certainly an issue in finding the patients, and for the patients who are BRAF mutant, but with a mutation other than V600E, we're a way away from finding therapies, certainly in lung cancer, which are even promising. So these are early days.
So if you had a patient with a V400 mutation, you wouldn't be giving them the...
Well, they wouldn't be eligible for this study, and I guess if the development of dabrafenib in particular continues along those lines, if it reaches registration, then it's going to be in a very defined subgroup of requiring a particular mutation. But that's I suppose, in some respects, the direction of travel. We've got a new generation of EGFR drugs now, which are specific for particular mutations. And all of this just begs the question is, how do we find our patients? And really the only answer to that is some form of broad multiplex testing or genome-wide analysis that we're going to have to implement eventually for our patients. And that's, of course, not going to be straightforward.
And that's going to be an important thing, given the very limited amount of tissue that we have with our lung cancer patients. We're already needing to do EGFR and ALK and possibly KRAS and BRAF. And presumably the only way we'll ever do this and have enough tissue is to be able to send off an array of testing.
Yeah. So literally salami slicing our tumour, as you imply, is even more impractical in lung cancer than most other solid tumours.
Yeah. And also, presumably, the more sophisticated that testing gets, the more likely it will be that it will need to be done centrally as opposed to being done in your local lab. That perhaps central testing of tumours in one large place might be the way forward.
Yes. That having been said, the mutation analysis and other forms of profiling are increasingly straightforward from a platform point of view, and the costs of these technologies are falling all of the time. So I think the main issue in the future is not going to be a moderate size centre being able to organise or afford that sort of testing. It's keeping up with the technology, and even the biggest centres are going to be struggling with built-in obsolescence as every 1 to 2 years the technology is transformed.
It was interesting to pick up on the plasma-based EGFR testing that was built in as an exploratory analysis on the PROSE trial in second-line non-small cell lung cancer. And it's worth reiterating the discussion that took place then, that EGFR unknown in tissue is not the same as EGFR wild type, and it's important to bear that in mind when thinking about prognosis.
And plasma DNA testing didn't have a sufficiently high sensitivity to be used routinely. It's quite a high sensitivity, but specificity, but the sensitivity was only around 60-65%.
So
currently not good enough.
Yeah, it's work in progress. It's not ready for prime time. But of course, the expectation is maybe we're getting a bit glib about technological advances, but I think the expectation is that it'll be there one day, and certainly in our tumour type, it's just it's built for us to be able to take a blood test and do a test for tumour DNA rather than having to put the patient through a tumour-targeted biopsy. It's got to be the dream and the aspiration. Thanks both.
Thank you.